Good morning and welcome to Biology with Ms. McCoy. And today what we're going to be doing is we're first going to review DNA structure, which we went over last class. Want to make sure everyone is comfortable with all of the information regarding DNA structure so that we can actually talk about how DNA is replicated. So first we want to talk about what actually is DNA. Well, first off, DNA stands for deoxyribonucleic acid. The deoxyribose is the actual sugar in DNA. It is a nucleic acid. So it is a double helix. It is made up of nucleotides. And we know that nucleotides or nucleic acids is one of our major macromolecules. And it is a double helix. It has three components. The three components are phosphate, sugar, and the nitrogenous base. So the three components, this is a basic structure here. This is not the chemical picture. This is just a structure of the three components. And what that is the phosphate and sugar make up the backbone or the sides, the supporting structure of the DNA. And the nitrogenous bases make up the rungs of that ladder or staircase. So, and they are paired in a certain way. Our adenine, thymine, guanine, and cytosine are our four bases. And they pair in such a way that adenosine always pairs with thymine, guanine always pairs with cytosine. And you're saying to me, well, why is that, Ms. McCoy? Why does it only pair that way? Well, first off, adenine and guanine are purines. They are a double ring structure. And cytosine and thymines are single rings. So you can imagine if I have a staircase and I have two double rings together, it's going to expand. And then the next rung would be small. So my staircase would not be a good stable structure. So our guanine are our purines, and our cytosine and thymine are our pyrimidines. One of my students yesterday, when we were talking about the bases, came up with a great little mnemonic for remembering how they pair. Apple, tree, car in the garage. So yes, adenosine and thymine always pair together, and guanine and cytosine. The other thing that is really important about our DNA structure, it is that it is anti-parallel. And you're saying, Ms. McCoy, what does that actually mean? Well, if you look at this side of the DNA, you can see that we go phosphate, sugar, phosphate, sugar, phosphate, sugar. And if you count the carbons on the sugar, we are going from the 5' to the 3' direction. Whereas if you see on this opposite side, can you see that my sugars are inverted or upside down? And now I am going from the 3' to the 5'. So they are mirrored images, but reversed. So they are anti-parallel. So when we go to replicate and duplicate our DNA, the rule is you read up. So you read from the 3 to 5 direction, but you write down. So you always write from the 5 to 3 direction. So you read up 3 to 5, write down 5 to 3. So that is some review of yesterday's DNA structure. Remember what our D in the DNA stands for is the deoxyribose. It's the sugar found in DNA, which we know from our discussion yesterday, it's different than RNA. RNA is just ribose. So, and then the three structures of our nucleotide is the phosphate sugar base. The phosphate and the sugar make up the backbone of the DNA, and our nitrogenous bases make up the rungs or steps of the ladder. Does anybody have any questions regarding our review? Because that is what we really went over really heavy last class, and we had a quiz and everybody did great when we reviewed the actual structure. So what I want to do today is go into how actual DNA is replicated. All right, now that we have talked about DNA structure and reviewed everything that we needed to know, we're going to talk about DNA replication. And we know that DNA replication takes part during which phase of the cell cycle? That's right, during the S phase. And we are going to now talk about duplication or replication of the DNA. Here is my DNA molecule I've drawn with my lovely highlighting markers. And imagine a zipper. And here is my zipper, and those nitrogenous bases are the teeth or the things that interconnect during my zipper. Helicase is my enzyme that runs down the length of the DNA and breaks those hydrogen bonds between the nitrogenous base pairs in order to open up the strands so that we can duplicate the DNA. We have our leading strand and our lagging strand. And the reason this is leading is because when we lay down those base pairs, remember, we read up and we write down, which means this strand is going to be able to be replicated continuously with no breaks and no gaps. Whereas my lagging strand, because of the fact that it has to write down, it's going to, and the way that it is opening up, we are going to have segments that are not completely copied on a continuous basis. It's going to be done in sections. And those sections are called Okazaki fragments, and we will get to that. So when on my leading strand, this one is going to be continuous. It is going to lay down continuously, whereas this one is going to write or lay down new base pairs in segments. So if we match our base pairs, so when we have our different base pairs on our leading strand, my new strand is going to be complementary to that. So can you see that I have one original DNA strand that is being paired up with a new strand? Again, my original and my new. So this is called semi-conservative. What that means is that my two new DNA molecules each will have one of the original strands and one new strand. So it is semi-conservative. Where the helicase separates the DNA, that is called the replication fork. And the other things that are playing a big role in our replication, one is polymerase. That is the enzyme that is working its way down and laying the complementary base pairs. And primase has to first lay down a little primer, and it's made out of RNA that basically tells the polymerase where to start and to start laying down new base pairs. But again, our helicase had to open up and create a replication fork in order for this process to happen. So if you were to break this down into 3 steps, you would say 1, the helicase unzips the DNA. 2, primase lays down a primer to tell polymerase where to start. And 3, the polymerase runs down the length of the DNA and adds complementary base pairs. Back to our Okazaki fragments. If this is discontinuous, we have segments. And so we have little missing pieces. So our polymerase and our primer has to keep going back and fixing those little spots that are missing. And that is what is meant by our Okazaki fragments. So yesterday and today we reviewed the important structure of DNA, and today we talked about the replication. We are going to now watch a short video or an animation on the actual replication process, and then we will have a group discussion, and we will go back over one more time and make sure that everybody is feeling really confident with DNA replication.